Thank you very much for having me there at this wonderful occasion, virtually, of course. I want to thank Robert Dudson and Peter Vanderspeck for their invitation, and I apologize to them and to the attendees that I cannot make it in person. Of course, I want to extend my hearty congratulations to the Erasmus Medical Center for their centennial. What an amazing accomplishment and milestone to reach a centennial. I wish I could participate in the festivities with you all in person today. But over the next 20 minutes, I'm going to be talking about this new direction of personalized medicine and precision medicine, and specifically focusing on big data in biomedicine. To me, I think big data in biomedicine is a most amazing platform for innovation discovery, and hopefully I can convince you of that as well. On this first slide, I just have to obviously give you my disclosures, and I just will simply say that I have started a bunch of companies, I consult for a bunch of companies. Perhaps you shouldn't believe another word I say over the next 20 minutes. But in all seriousness, I'm most proud of, at the bottom right, you see a list of the companies started by my students. More than half of my graduate students now have started a company. And they're able to do this with this most amazing platform in the world. This platform of open data, open big data. Where does this open data come from? Why can we even discuss big data in biomedicine? On this slide here, you see what is commonly known as a gene expression microarray. On the left, a particular microarray from a company called Affymetrix, actually just down the road here in Silicon Valley. And this chip on the left here can quantitate or measure every gene in the genome, specifically looking at the RNA. So you can use these for DNA. Here I'm specifically talking about the RNA, the blueprints on their way to coding for proteins. And one chip, it kind of looks like a computer chip, as you all know, by design almost. I mean, Intel is right down the road as well from here. And that one chip lets us quantitate the entire human genome for a couple $100. Now, when we get tired of measuring them one by one, we can measure them 96 at a time. So on the right, you see the 96 well format for these arrays. Each well, each little square there is its own little microarray. Now, this is an amazing technology, but can we still believe it's been more than 15 years since we've had these microarrays? And companies like Affymetrix, Illumina, Agilent, and many others now make these arrays, as well as homegrown arrays from universities. What I still think is the most amazing accomplishment in biomedical science is that we make scientists share this data today on the internet. Now, in the beginning, it was hard. My first experiment back in 1998, 1999 composed only 60 arrays, precious arrays. And people started writing papers, as we did, as did others in the Netherlands and all across Europe, about arrays and how they could be used to find differences in genes between disease conditions and diabetes and cancer, and bad cancer versus not so bad cancer. But the journals and the editors were the first to say, we are getting these papers, but we need to start to get the data and release that data to the public. And those requirements started from the publishers and then expanded to the funding agencies. So the United States National Institutes of Health and the Wellcome Trust and many others now require this data to be deposited on the internet. So fast forward to today, or specifically last year, we hit a milestone. In August 2012, as you see in this slide, special article in Nature indicated that we reached 1 million of those microarrays publicly available. And you can see the growth in the graph here. And it's exponential growth, exponentially growing, starting at 0 in 2000 all the way up to 1 million. About 3/4 of that in the United States repository, another quarter in the European repository, Array Express. Now, this is gene expression microarrays, but this is just symbolic of many types of high-throughput measurements in life science data. This is one type of measurement here. And to be very specific, on this slide, you see the specific website. So here I'm showing you the NCBI-GEO, Gene Expression Omnibus, in the back, and the Array Express in the front. And actually, you can see 1 million was last year. We're already up to 1.25, 1.25 million arrays this year. We've already added another 250,000 arrays since that article came out last year. Now, this is just an amazing concept, but it's hard to kind of understand all these millions and thousands, what they actually mean. So I've said this in talks many times before. I'll say it in front of you here today. This is the power of public open data. Even a high school child today that needs to do a science fair project, she can go to any one of these two websites I'm showing you here, type in the word or phrase breast cancer, if she wants to start a science fair project on breast cancer. And here on this slide, you see that she can download and find nearly 40,000 samples of breast cancer. 40,000 samples of breast cancer. About as easily as she could find a song on iTunes or a music video today. 40,000 samples of breast cancer a high school child can actually get for their science fair. What's so magical about 40,000 samples? If it isn't obvious, you'll start to realize that's more samples available to this high school child than any breast cancer researcher has. Because, of course, that high school child has what any one breast cancer researcher puts in the international repository, plus all the competitors. So anyone who can use this big open data now has more samples than anyone contributing data into these repositories. Now, 40,000 samples is pretty impressive, but actually the number I like to actually discuss is a little bit above that. You see above 39,000 is 1,500. So more than 1,500 labs have contributed their data into this repository. Now, 1,500 is, again, hard to imagine what that means. Let me present it to you this way. Let's say I want to start a brand new effort to study breast cancer today. And I want to get my friends on the internet to help me study breast cancer. So the kids today would call that crowdsourcing. So I want to crowdsource research in breast cancer. And let me write a proposal to you. Okay, you're going to review my grant. I'm going to get 1,500 of the best labs around the world to get their best patients with and without breast cancer, to use the best technologies they have at their disposal, to collect all this data and give it to me for free. Now, if I were to propose this in a forward direction, you would laugh me out of the room. You would say, how on earth can you get 1,500 labs to share data with you for free? Yet, I'm showing you on the website here, there are already 1,500 labs that share data with you. So when I think about public big data like this, or open data, I actually think of it, another way to phrase that is retroactive crowdsourcing. What does that mean? You can get 1,500 labs working on breast cancer. Those 1,500 labs are helping you with breast cancer research, and they don't even know it. And those 1,500 labs are helping you with breast cancer, and you don't even know it. Yet, there it is, 1,500 labs, 40,000 samples. And if for some reason that's not enough for you, wait another 2 or 3 years, and it'll reach 80,000 samples. Because just by sitting here, the data continues to grow. What an amazing world. What an amazing technology and platform we have for innovation already. Now, some of you might doubt, how can a high school child actually even do this? So on the next slide here, I'm just going to show you one example of a high school child. Her name is Brittany Wenger. I've actually not met her. She's from Florida. And she just happened 2 years ago to win the top prize, the grand prize, in the Google Science Fair competition. So Google gives out a prize for the best high school student in their science project. On the left, you can see that she actually built a neural network, a machine learning classifier, on breast cancer using publicly available data. Now, she also used histopath images and things like that. And then this year, she said, I guess if that wasn't enough, this year she built another diagnostic for leukemia using public data. What an amazing world that even a high school child can do this. And of course, what I'm trying to say here is if a high school child can do this, perhaps the rest of us in biomedical research can do this too. So my first big grant at Stanford from NIH was, let's just go get not just breast cancer, but every human disease studied by microarrays. On this slide, you see the kind of end product of that work in the first 5 years. We built a grid of all the different samples. On the rows here, going top to bottom, you see various different diseases like leprosy, HIV. Of course, we have a bunch of cancers. And the columns from left to right are all the different genes, 20,000 genes here. And to make a long story short, we can normalize, we can scale the measurements, we know how to compare one type of microarray platform to another, we know how to find these samples using text mining. All of those are shown in the references here. You can find any of those papers, all the different technologies. But in the end, we have more than 300 different diseases and growing, all illustrating different diseases studied by microarrays already to date. And we already start to get ideas here. You know, these two diseases maybe are similar to each other. At a molecular level, boy, the same genes are going up and down, but the doctors aren't the same. The doctors don't even think of these as being the same disease. But where we really start to get excited is on this slide, that as we're building this collection of diseases, others are starting to build similar collections of drugs. So on the left, you just get an illustration of this. On the left, we have samples from healthy patients and samples from those with disease. So with disease, without disease. With disease, without disease. And on the right, investigators like Todd Golub at the Broad Institute, Justin Lamb, and actually many others now are studying drugs. And they take cells, initially 1 cell line, 3 cell lines, now up to 15, 20 cell lines, and they're studying them with drug, without drug, with drug, without drug. And all of a sudden we realize both of these types of repositories are publicly available. Why don't we put those two together? And the point of putting those two together is to try to match a drug or a drug candidate with a disease. Why can't we try to match those two? And to make the statistics very simple, I'm going to use my arms here. So if you can imagine, I have a disease where this gene goes up and this gene goes down. And now, from the other side, I have a drug that can make this gene go down and this gene go up. Maybe there's a perfect match between this drug and this disease. Now, these are all hypothetical, and these can be tested. But where we really start to get excited to move drug discovery along faster is not just to find brand new drugs, but to find new uses of existing drugs. Drugs that might already be in the clinic for one reason or another. So the idea here is called drug repositioning. And of course, drug repositioning has a well-known history, going back to famous drugs that we have famous cardiac drugs that give us interesting side effects, like hair growth. That, of course, is minoxidil today. Of course, everyone knows the famous cardiac drug that had a very interesting side effect, which today we know as Viagra. Those are the brand names, of course. And Viagra has actually done a double switch. Some people might know that Viagra is also available as Revatio, the same molecule, but a different brand name for pulmonary hypertension. But you know what? Instead of finding these by accident, let's start to find them on purpose using public big data. So we take our diseases, take our drugs, we start to get hundreds, thousands of such possible combinations. Now, one of the first ones we chased down is actually a very interesting finding, looking at an anti-seizure drug, epilepsy drug known as topiramate, we predict works on Crohn's disease or ulcerative colitis. And on this slide, you see my famous rat colonoscopies. If you have to think about it for a moment, rat and colonoscopy typically don't go well with each other. And I have a whole story about how we can outsource the mouse models and the rat models, indeed what we did here. And you can see some evidence that on the left, or in this first video, you see what a normal rat colonoscopy looks like. The rat is obviously still alive when you can get the colonoscopy. When you inject or introduce a particular chemical called TNBS, it's the model, one model of inflammatory bowel disease. You see redness and inflammation. And then you can see that we have another rat given the TNBS, but also given topiramate, and you can see that it's better. And I don't have time to show you the histopath images and all the rest. But this gives us an idea now that we can start to move drugs, computationally predicted, but test them in animal models, and indeed find new uses for drugs. This particular molecule, topiramate, is already finding other uses out there, including obesity and others, but is already available in generic form. So in other words, pennies per pill. Now we've done maybe 10 of these in the lab so far, okay? We're very excited about this. But this next one is the one that excites me the most. Why am I showing you this one? So here, this is a very close collaboration with Julian Saj, Nadine Janchan. Computational work was done by Joel Dudley, as was the previous work, along with Marina Sirota. And this one's exciting to us because this one is a mouse model that gets small cell lung cancer. So humans typically have to smoke to get small cell lung cancer, though it's a neuroendocrine tumor. You can get it without smoking. This mouse, if you knock out these 3 genes illustrated on top right here, this mouse gets its own small cell lung cancer. You have to wait weeks for this experiment to start, and then when the mouse gets the lung cancer, you can see the nodules on the top left there. Now on the right here is the same type of mouse getting small cell lung cancer treated with an antidepressant. Okay, this is a psychiatric drug. It's a class of drugs known as tricyclic antidepressants. This one is known as dizipramine and imipramine. These are in the class of drugs. And dizipramine, imipramine, and tricyclic antidepressants are typically not used anymore in humans. They have some annoying side effects, arrhythmias, they can make you sleepy. Nothing as bad as having lung cancer, though. And on the right, you can see that this mouse doesn't have the lung cancer anymore. The small cell lung cancer has disappeared. It's melted away. Now, this is a mouse model. But why does this story excite me? Because we start with public data, public open data. We get our computational prediction for the disease and the drug. We get it tested in cell lines. I'm not even going to show you that. We get it tested in this animal model. And then 15 months later, 15 months after the computer prediction, we get the ethics, the Institutional Review Board, IRB approval, and we've already launched this in a human clinical trial. 15 months from computer prediction to trial. I don't know if this drug is going to work in trials. The trial is being run by an independent team, run by Joel Neal. I'm financially connected to this drug, so I cannot be part of this trial. So I don't know if this drug is going to work or not, but to me, this is the most amazing next step. Not just playing with public big data, but to take it to patients in real clinical trials. And not 10 years and $1 billion later, but perhaps getting drugs to patients that much sooner. That's why I'm excited about this finding. And of course, in the Stanford way, Stanford tradition, we launch a company on this. So we've already launched a company called Numedi. They've already gotten their Series A financing about 6 months ago, about $3 million in outside investors around here, Sand Hill Road and others. And they already got their first pharmaceutical company deal. It turns out that there are pharmaceutical companies that work on specialty pharmaceuticals that don't have much of a research and development arm, but they've already got their first deal, and they're working on several others. So there is a market for these. A company like Numedi is going to be able to take public big data and come up with these kind of new uses for drugs. They can work on the intellectual property, the reformulation, but this could be a new type of pharmaceutical company. Maybe it doesn't take the drug all the way to patients, but at least can get it to develop to such a stage and maybe get pharmaceutical partners to take it the rest of the way. In the last few minutes, I want to talk about the other big source of public data, of public big data, and that's coming from genome projects. Obviously, you know that the Human Genome Project was finished around 2003. It took about 13 years to get the Genome Project done and about $3 billion, and it was done in centers like this. This is a picture from the Whitehead Institute in Cambridge, Massachusetts. And you can see a bunch of robots, a bunch of people picking clones, running sequencers. And obviously you know that in the subsequent 10 years, the price of genome sequencing has plummeted down to thousands of dollars, maybe even $1,500. In some cases, it doesn't take 13 years, but we can get it done in 27 hours, so maybe a genome in a day. And on this slide, you see that the genomes are increasingly going to be done not just in tabletop sequencers, but maybe even sequencers running off of the USB stick attached to your computer. So this is a company known as Oxford Biosciences. They may or may not come up with a product someday, but they're at least showing it off at their meetings. And we can all imagine that we might get our sequences done with a device like this. It's a no-brainer. We're going to have an impact in medicine. Now, this is a personal big data problem. Personal big data. What do I mean by that? This is an issue where you are going to get the 6 billion base pairs, 3 billion from your mom, 3 billion from your dad. What we learned is that this is no longer going to be a sequencing issue anymore. That's not the hard part. The hard part is going to be in interpreting these sequences. So we started an effort 6 years ago in my lab because I knew someday a patient was going to appear at Stanford with their genome. We started an effort to use published big data, in other words, all the data stored in publications, and there are many publications from the genetics community, to help interpret personal big data, all the genome sequences in you. It turned out there was no master list of all the different sequences out there that had been found to alter one's risk for disease. These SNPs are differences in your spelling in your DNA. What we realized is we're going to have to reread every single paper in human disease genetics. And in the past 6 years, we've now read close to 20,000 publications. We've reread every one of those publications, writing down and curating every single SNP associated with every single disease. And to make a long story short, we probably have one of the best lists of human disease SNPs in the world. And here you can see what 1,000 papers look like. Imagine 20 of these. These are actually the front pages of 1,000 of the papers we've read. And what we can do, because we've written down now every single odds ratio and p-value and risk assessment, every disease using standardized vocabularies, we can start to make a personal risk prediction. So on this slide you see what we did with one of the first patients to show up with their genome, Steve Quake. He happens to be a bioengineering faculty member, but showed up at Stanford because he had a question about his own health, his own risk of sudden death. And we can make what we call a riskogram here. This was published in The Lancet in 2010. You look up this paper. The black arrows indicate different diseases and what we thought one's risk was for those diseases using prior probabilities and epidemiology. And the red lines indicate new predicted higher risk. The blue is protection. Those are how the risk assessment has changed given those genomes. Now this is neat. When we can do it for one by one, we've done this on families. We've done this for children and adults with interesting disorders or rare disorders. On this slide, I just want to point out, this publication came out earlier this year in PLOS Genetics. This is what 1,000 people look like. So literally what you're seeing here are now 1,000 people, each with a genetic assessment. These happen to be native populations. So they might be Aboriginal tribes, they might be Native Americans. This public data set of 1,000, this is looking at just one disease, type 2 diabetes. And what we start to realize is that one's genetic risk for diseases is not equal across the world. Europeans, Northern Europeans, have different genetic risk than Southern Europeans. Certainly those in the Americas and Africans and East Asians and Islanders all have different baseline genetic risk for different disorders. At least some genetic disorders, including type 2 diabetes. So we don't all start with an equal genetic risk. On top of that, we certainly have unequal environmental risk. Certainly in the United States, we have this interesting restaurant called Cheesecake Factory. You can imagine a factory giving you cheesecake and equal-sized portions, and we have huge portion sizes. Obviously a big problem with obesity in the United States, but also a problem now increasingly a problem in India and China as well. So genetics, a different environment is different. And this is going to be how complex disease prediction is going to be in the future. But we have to start. We have to make a step. And of course, Stanford tradition is to spin this out into a company. So I'll just make a note that we have a company called Personalis now, which has licensed this database as well as others from Stanford, and has already started to get investor financing, incredible investor financing, including the UK investor known as Abingworth involved with this company. And obviously, they've already gotten their first major contract is to do what's called a Million Veterans Program. So to study at least the first couple thousand of the million veterans that are treated by the single-payer healthcare system known as the Veterans Administration in the United States. To study them longitudinally, to look at electronic health records, but obviously to resolve that or compare that to their genetic predictions. It's here, too, where I do want to bring up the potential of collaborations, both from the company side as well as the academic side. Peter and I have known each other for nearly 10 years, and I do hope that we continue to grow collaboration. But actually actively collaborating right now are Ling Oy, Henning Timir, and Andrea Utlingen, who are collaborators with the Rotterdam Heart Study. And we're hopeful to use this database of these complex SNPs, these misspellings in the DNA, to try to predict or retrospectively predict if we can find out how and why people die of various disorders, with the hope of trying to prevent it in future people, future patients. Now, this is looking at the complex disease side of the genome. But an amazing story is also there for the rare disease and the rare children. On this slide, I just want to point out another story. This is done by investigators in Milwaukee, in Wisconsin, in the United States. This child, Nicholas Volker, they say is the first child to be saved with DNA sequencing. And to make a long story short, for 5 years of his life, since birth, he was losing parts of his colon. He was actually losing his bowels. Surgery after surgery. Parents losing hope. Patients losing hope. The families losing hope. And they said in a last-ditch effort, let's get his genome sequenced. So they sequence his genome, sequence his parents. They realize he has a novel variant in the gene that's expressed in the lymphoid tissue, and this prompts them to do a stem cell transplantation in this child. Extremely high risk, but successful. And now this child goes to school, eats regular food, This is the first child to be saved with DNA sequencing. It's an amazing story. There's a Pulitzer Prize winning news story around it. You can Google him up. I recommend you see this story so you know what can be done with big data in this personal type of way. On this next slide, though, I think of as the next big revolution in open data. So about a year ago, I was asked to take over this new project called IMPORT. This is from the National Institute of Allergy and Infectious Disease, NIAID, one of the larger institutes of NIH in the United States. And this was formed about 5 years ago, this repository called IMPORT, to handle immunology measurements. For example, flow cytometry files, ELISA spots, flu measurements, influenza measurements. And I took this over. We kind of have launched on this. And when I took it over, I realized that it isn't just about another type of molecule. What IMPORT has is actually the next big revolution in open data. And that is releasing raw clinical trials data to the public. Open clinical trials data. I think this is the next big revolution. I think journals are starting to not be happy to see that clinical trials data locked up, especially when, for example, recommendations about Tamiflu and other types of drugs. There's a big fight between British Medical Journal, BMJ, and Tamiflu, which seems to be resolved now. But if governments spend hundreds of millions of dollars stockpiling medications and public policies change because of medications and these papers, perhaps some of this data, individual raw clinical trials data, should be released to the public for transparency. Now, I'm not talking about making identifiable data. This is de-identified data. But I think clinical trials data is going to be the next big revolution. It's going to start from the journals, then it's going to move to the funding agencies. I think we're going to see a new world of, imagine what we can do with clinical trials data. Maybe putting 10 trials together, maybe doing meta trials. I see a new world for this high precision type of measurement. But of course, that's just the beginning. We have so much other type of data out there, including the Cancer Genome Atlas and equivalent projects from the Europeans. We have ENCODE looking at different pieces of DNA. We have EPIC and Cerner, the electronic health record companies, and the back-end data that's being collected on humans and patients. We have pharmacogenomics data, the side effect data for MedWatch, as well as the European Sentinel projects. Of course, entire countries getting tens of thousands, hundreds of thousands of patients sequenced, and on and on. So in the end, what is big data in biomedicine? What is big data in biomedicine? Is it about algorithms? It's about programmers and parameters, databases? Is it cloud computing, Hadoop? Is it about high-performance computers, mobile, e-health? I actually think big data in biomedicine is about none of these things. To me, big data in biomedicine is about predicting the disease before it strikes, deciphering those rare diseases that just defy experts, finding drugs for diseases that just aren't getting much attention today, making sure we do the right, safe, cost-effective thing for patients. And I think open big data is the biggest, the most amazing platform for biomedical innovation and entrepreneurship. And I can sum all that up with one word: hope. I think big data biomedicine is all about delivering hope to patients, to families, to healthcare providers, to researchers. Hope for a better world for medicine. So I've got to put in a plug for my own meeting. We have a meeting here at Stanford. This is the second meeting we'll have for Big Data Biomedicine. It's in May. I have to thank a long list of collaborators, as you see on this slide, specifically Alejandro Sricardero and Julian Sage on the mouse models for cancer, Jay Pashrika for the rat models of Crohn's disease, inflammatory bowel disease, as well as Ewan Ashley, Steve Quake, Russ Altman, Mike Snyder, Carlos Bustamante on the human genetics work. And I get generous endowment support from Lucille Packard Foundation for Children's Health, NIH, the National Institutes of Health, March of Dimes, Hewlett Packard, stem cell propositions here in my state of California, disease foundations that want us to find new drugs for their diseases. I always thank my admin and tech staff. I'd never get a grant or paper out the door without them. And I always thank my daughter who inspires my work, Kimayani Butte at the bottom, and my wife, Tharangini Deshpande, who is a molecular biologist biochemist who reads every major paper and grant that comes out the door and starts new companies with me and tolerates me giving talks all over the world like this. Thank you very much.